Hello and a warm welcome to our poster. My name is Annette Raffin and I wanted to give you a quick walkthrough of our work on how we can exploit not only plant roots but plant architecture to help understand community-led mechanisms of root influence on soil structure. We're particularly interested in ley grasslands. Why? Well, because they are a key time in agriculture when soil can have a rest from the intensity of crop production. This is particularly relevant in terms of restoring soil structure, and we're looking at how we can use plant roots to drive this structural regeneration. And not only plant root traits on their own, but plant root traits together as a community, working together. Plant roots work hard to get the resources required that they need for growth, including obtaining nutrients and water. What's more, they alter their root and shoot architecture and behaviour depending on the availability of the nutrient. Because the plant architecture changes, we hypothesize that the soil structure will also change. It gets even more interesting when we consider the community aspect, because plants are competitive for nutrients and alter their root architecture further when there's someone else around to compete with. So this begs the question, can we manipulate a comparatively immobile nutrient like phosphorus to stimulate changes in plant architecture to alter soil structure? We selected two grassland plants with very different root characteristics and phosphorus responses. These were Timothy, a fine rooted grass, which being a grass is highly competitive, and had a high shoot response to P. This means that the root response may be similar, irrelevant of phosphorus concentration, but the drying effect from the shoots might be very different. The other species was chicory, which has a dominant taproot and is relatively unresponsive to phosphorus, plus has a lower competitive ability. We suspect that under low phosphorus conditions, roots have to work harder locally to extract phosphorus for the given allocation to root architecture. On the other hand, under high phosphorus, the impact is more extensive, as phosphorus is no longer a limitation, and larger distant effects such as increased drying tend to take over. By stimulating competition and altering species, we can manipulate these effects, and including impacts such as depth. The experiment is split up into two parts. The results of part A are presented briefly here, whilst part B is ongoing. We plan on using root inclusion and exclusion cores to separate local root impacts from these distant root impacts, plus they'll give us intact specimens to study soil architecture. This diagram highlights the proposed mechanisms and the different root types of the two plant species. Under low phosphorus conditions along the top here, we expect that there will be limited effects on the macrostructure, but more intense local effects on the microstructure. Conversely, under high phosphorus conditions, we'd expect more extensive effects at the macrostructure level and less impacts on the microstructure. From the first part, we confirmed our expectations from the literature that phosphorus had a limited impact on root architecture of the two species, but that there was still a response in leaf area. All compositions increased their leaf area relative to their root surface area. There were indications that root length density was greater in the mixed compositions with phosphorus application, but this wasn't significant. But this will be of great interest once we move on to the next stage and work with older plants. There were no significant drying effects of the plants at this size, though. Yeah, we did still see evidence of structural effects with phosphorus decreasing macroporosity. There were also some quite interesting shifts in the pore structure, such as these radiating cracks created by chicory roots, seen here in the X-ray CT scans. This is presumably as they dry out the soil. There was some evidence that Timothy generated larger pores and chicory smaller ones, but the suspicion is that Timothy may homogenize pore size distribution more, whereas chicory will tend to polarize the pore size distribution. For the second part, we're basically expanding on part A by extending the scope of the study to identify whether this proposed mechanism of where phosphorus will impact structure occurs. So what we'd like you to take home is that phosphorus has the potential to influence soil structure through altering plant architecture as a whole and not just through manipulating roots. This is important to note because above ground vegetation may ultimately be the greater driver in soil structure, not roots. Furthermore, we hypothesize that low phosphorus conditions drive microstructural impacts, whereas high phosphorus conditions drive macrostructural impacts, but that these effects will likely be dependent on root type and composition, along with above ground plant properties. Thank you for taking the time to listen to this presentation. I wouldn't be here without the great help and support of the Soil Biophysics Group at the University of Aberdeen, and also to my funder, the NERC Quadrat DTP. If you have any questions or want to get in touch, join us at the poster or scan the QR code here and that'll tell you everything you need to know.